Hi, my name is Sam Cooper, and I'm one of the co-founders of Phenomic AI, also the CEO. And our mission at Phenomic is to deliver novel therapeutics against non-small cell lung cancer to the clinic. So let's outline the problem. Lung cancer is a global issue. 1.7 million people die of lung cancer every single year, and in 90% of the cases where treatment fails, this is because chemotherapy resistance develops. This highlights that there's still this massive and urgent need for new therapies to combat resistance and help reduce the number of deaths from lung cancer. What we at Phenomic aim to do is deliver new therapies, new diagnostics to the market to enable us to combat chemotherapy resistance. Much of this problem stems from the fact that non-small cell lung cancers are highly heterogeneous. Within a tumour, many different types of different sort of genetic subtypes of cells exist, and some of these are resistant to chemotherapy. At Phenomic AI, we're looking for therapies that will go alongside traditional chemotherapies and target some of these major subpopulations that become resistant. Together, we believe this will reduce the likelihood that resistance occurs and if it doesn't increase overall survival, at least increase the length of healthy life that patients with lung cancer can enjoy. Over my PhD, I was working with a postdoc where we identified a subpopulation of lung cancer cells that are chemotherapy resistant. Critically, we uncovered the genetic target that is responsible for this resistant subpopulation. If we get rid of this target, we sensitize these cells versus other cells to chemotherapy. However, there's a big problem. This target is undruggable. So what do we do? Oh, and when we say undruggable, what we mean is basically it interacts through protein-protein interactions and interfaces that we can't just bind drugs to. So what do we do? Well, it turns out that this subpopulation actually just demonstrates, quite specific phenotypic properties. And what this will enables us to do is identify what are called antibody therapeutics that selectively bind this subpopulation. By selectively binding this subpopulation, we can then tune these antibodies to recruit our body's own immune system to attack these non-small cell lung cancers, these chemo-resistant subpopulations in non-small cell lung cancer. But the problem is we don't know exactly which targets are, selected for this distinct population. So what we're going to employ at Phenomic is, sort of a really novel and very underused technique called phenotypic discovery. Phenotypic discovery is very active in sort of small molecule spaces when you're looking for small molecules, but it's almost never used for antibody discovery approaches. So what we're aiming to do is use these phenotypic approaches to identify novel targets that are selective towards this subpopulation, and then use these antibodies to recruit our own immune system to target these chemotherapy-resistant cells in lung cancers. The real technical challenge that emerges here is finding the right antibody that's hitting the target that's selected. In an antibody library, there may be up to 10 billion or maybe even 100 billion antibodies. So how can we find which ones are selective to this chemotherapy-resistant subpopulation and either select or modulate this population, just kill it outright? There's a really great strategy called panning, which allows us to basically use genetics and evolution to enrich antibodies for things which are most likely to be selective. But the problem is, if we still then have 1,000 antibodies which may or may not be selective, and we need to figure out which one of these 1,000 sort of enriched antibodies are working. And this is really an unsolved challenge, and why most people don't use phenotypic screening approaches for antibody discovery. But we're Phenomic AI, and we have developed deep learning solutions that we believe will allow us to solve this problem. Specifically, by using high throughput microscopy and imaging, we can very accurately identify specific mechanisms of action or functional activity that diverges from control populations. Additionally, this also lets us, immunofluorescently label certain things or label certain sort of antibodies in our population of cells with sort of reporters and detect which are selectively binding this subpopulation. However, when we do this, the problem is with doing this, high content imaging is we can get great images and see what effect it is. But when we're testing 1,000 antibodies, we literally can't process all of these by eye, or at least accurately enough to say that this is having a meaningful effect versus control population. So this is where our proprietary deep learning platform comes in, and there's some great data here in this slide showing where we've used it already in a proof of concept study to demonstrate how we can pull out different mechanisms of action of compounds using no prior knowledge. So in this graph, we basically can demonstrate that we can find functional compounds against certain types of cells and demonstrate, we can identify cytotoxicity without having ever, understood what this condition will look like before. And so using this approach, we hope to be able to, identify which of these 1,000 antibodies that we have enriched against the subpopulation are most selective for these cells that we can then use to bring towards the clinic and eventually develop therapies from. So at the moment, we have $2 million in funding, and we're using this to identify a set of antibody hits that are going to be active against this assay. We also want to demonstrate that our platform is applicable to different diseases, so we're working against pulmonary fibrosis in a related sort of assay as well. We're then looking to take our antibody hits towards the clinic through doing preclinical development, selecting leads and patenting these. And from the patents, this is where the real defensible IP for in the long term, sort of value in our company will come from. We'll then perform IND enabling studies that will then basically allow us to then submit an IND application and start to bring these into phase 1 trials and first-in-man trials. As we're developing antibodies, the route towards the clinic is reasonably streamlined now. We'll also be doing antibody screening against this pulmonary fibrosis assay that we're developing now. We'll then look over after this sort of period of time, we'll then look to raise increasingly large amounts of money as we bring these antibody therapeutics through phase 1 and phase 2 trials, as well as expanding our platform to include new indications and developing our pulmonary fibrosis hits as well. There's 2 co-founders at the company, myself, Sam Cooper, and my co-founder as well, a guy called Oren Kraus, who was one of the first researchers to apply deep learning to drug discovery. With a scientific advisory board, which includes both AI expertise and high-content screening, as well as sort of more serious pharmaceutical expertise, with David Swinney, who's developed drugs through to the clinic before, and Joel Dudley, who's previously founded companies that are looking to use AI to reposition different drugs around different drugs. We also have expertise on the disease front with Boris Hintz, who's a world leader in fibrosis research. We've got a team of 11 people now, and who've previously got experience in developing therapeutic antibodies with some patents, as well as now a machine learning researcher who's had industry experience as well. So together, our vision at Phenomic is to develop one of the world's first phenotypic pipelines for discovering novel antibodies. This will be driven by deep learning, and we hope will allow us to bring multiple new therapeutics to clinic, both against lung cancer and other diseases that we're currently working to try and develop assays for as well. So thanks for listening. I would really love to present at this challenge in Shanghai.